Our protocol improves both throughput and time efficiency for obtaining RT-qPCR data directly from single worm or small bulk samples without the need for RNA isolation.
Using this protocol, over 9,000 RT-qPCR results can be obtained in just 2 days of bench work, a process that would typically take about 5 weeks using standard 96-well qPCR.
This protocol provides a fast, robust, and highly sensitive RT-qPCR assay in C. elegans, which is ideal for monitoring the inter-individual variability in the gene expression between isogenic worms.
Begin by picking the worms from their bacterial lawn onto a fresh unseeded nematode growth medium plate and allowing the worms to move around the plate for 5 minutes. While the worms are acclimating, in an RNase-free hood, mix 10 microliters of lysis buffer with 1 to 100 DNase-1 per sample, and place the lid of a PCR strip upside down on the platform of a dissecting scope. Next, add 10 microliters of the lysis mix to domed PCR tube caps, and scoop the worms from the plate to avoid transferring bacteria into each slot of the lid. When all of the worms have been transferred, close the caps, and spin the tubes for 5 seconds before placing the tubes in a Dewar flask of liquid nitrogen. After the last 5 seconds, thaw the tubes in a 40°C water bath before repeating the freeze-thaw procedure 9 more times. After the last freeze-thaw cycle, mix the lysed samples on a thermal mixer for 20 to 30 minutes at 4°C and approximately 1800 revolutions per minute. At the end of the mixing incubation,